9 people on the call right now. Why don't we get started? I don't have the list. This is X9 for the students that you have as part of the program, but I'm assuming most of them are here. I don't recall everybody's name over the past few weeks, but we'll get started and people can join. I'll just monitor the waiting room as well. So students, you probably again have the bio in front of you. Some of you may have read it, but I would just like to introduce Kelsey Keith. Is joining us today from the Coriell Institute in Camden, New Jersey. I got partnered up with Coriell. We've been doing a video project together to highlight and to provide career awareness at places like Coriell. She, I'm assuming, might tell you more about the research facility there. And Kelsey actually was one of the people that were interviewed for that video project. So she's joined us on a couple different occasions now. And she holds a BS in biology and a master's in bioinformatics, and is an associate bioinformatics scientist at Coriell Institute for Medical Research in Camden. I'm excited because I don't know a ton about bioinformatics myself, so I'm excited to hear what Kelsey has to say. And I'm hoping that she might also, and if she doesn't touch on this, maybe I can encourage her to. But it sounds like from her bio that she runs a bioinformatics summer experience, a new remote internship option for undergraduate students that she started during the pandemic in 2020. So maybe you could add a little bit of something about that if you hadn't planned on that, because maybe these students in their upcoming academic year, when they enter into higher education, they could also look at that. Yeah, so thank you, Kelsey, for joining us.
Oh yeah, thanks for having me. It's a pleasure to be here. I'm always happy to come in and talk to aspiring scientists, aspiring doctors. Like, this isn't something I had in high school and I wished I did. So I'm very happy to come and give you a talk today. Yeah, so I have a PowerPoint presentation for to show today. And I wasn't going to talk about the bioinformatics research experience, but I can throw some in at the end. But please, if you have a question, please interrupt me and just ask it, unmute yourself. This will be so much more fun if we're talking rather than me just sort of giving a lecture in bioinformatics. I'll try and keep an eye on the chat, and there's a couple of pausing spots in my presentation, so I'll pause for comments, but please just unmute yourself and ask a question, especially because it's a complicated topic, so I had to put a lot of explanation in. So if you don't understand something, please ask. Okay. So just to start off with, what is bioinformatics? It's also known as computational biology, so you'll see that as well. So the way I would describe it is you study biology using computers to analyze data. So it's the intersection of biology and math and computer science. And by math, I mostly mean statistics. So basically the job is sitting at a computer and analyzing data. And I just want to mention that there's two major branches. There's structural biology, which I don't do. I'm not a structural biologist. And there's genomics and epigenetics, which I'll explain more in a minute. And that's what I do, so that's what I'm going to talk about today. But there's many, many different subfields. Okay. So just want to give a really brief history of bioinformatics. It's a very new field. It's been around at most from the 1950s, if you consider it going back that far. So in the 1950s, people started looking at protein sequences. And you have to, remember, in the 50s, that's when we discovered that DNA held the code for genetic information for life. So this is really, really basic stuff. We just barely are starting to understand how a cell works and a cell stores information. So you're looking at So if you remember, like the central theory of biology is the DNA holds the genetic information, and then it's copied with RNA and then translated into proteins that do the work. So we couldn't even really look at DNA, so it started with protein sequences, although later they get to DNA. So the idea was you could represent the amino acids that make up proteins as single letters, and then you could compare the sequences. And so by comparing sequences between different people or between different species, you can identify interesting information about the protein. So for example, if you compare protein sequences from people who have a disease and people who don't have a disease, you can maybe figure out what is the problem with the person with the disease protein. Like, what's the part that's going wrong? Or if you compare across species, so for example, if you take human protein, you compare it to a chimpanzee protein and a gorilla protein and a bonobo protein, you can figure out what are the most essential parts of the protein. So what are if the parts of the protein stay the same across species, those are the things that make it function, and they're important and we want to study them. And this is still done today. It's more advanced than it was in the 1950s, but it's still like uses a lot of the techniques that were developed then. And an example was they looking at people with HIV and people who were protected against HIV. They identified a mutation in a gene called CCR5 that protects you from getting infected. So this is still some people do this. This isn't what I do though. So the other big change in bioinformatics came with the Human Genome Project in the 1990s and early 2000s. So this is an enormous deal. So before the 1990s, we didn't know what the sequence of any genome was really. So this was a huge investment, primarily in the US, but also in Europe and a couple of Asian countries, that hugely advanced sequencing technology. We figured out how to do it much faster and better and cheaper, and we sequenced the human genome, and then everyone had a common reference they could work off of. So now, instead of looking at individual proteins or individual genes, you can compare all the DNA. And you can do genome-wide analysis, and that's what I do. Like, we sequence samples, and I compare a genome-wide something between samples. And for like a really cool example of what you can do with this, so this is a map of Europe, and people sequenced hundreds and hundreds and hundreds of genomes and looked at their genomes. And by looking at your genome, you can see their ancestry. You can see where their ancestors came from. So like you can see like people in like this orange, they were from the United Kingdom. You can see people who were from France, people who were from Eastern Europe, people who were from Italy. So your ancestry is in your genome. Okay.
So, Kelsey, sorry to interrupt. Quick question. I know this isn't necessarily the work that you do, but just to kind of put this into, or try to put my thoughts into a real world context, is this like the 23andMe type of service that you can get your genome sequenced and you can look at your ancestry and things like that?
Yes, this image is actually from 23andMe.
Okay, yeah. Cool.
Yeah, perfect. Okay, so how does this work? So you have your DNA, you have the 4 bases. So there's no, it's not working. So basically is you extract DNA from some sort of cells. It could be a sample a patient donated, it could be a model organism that you sacrificed, it could be cells that you've cultured in a lab. And there's then you do shotgun sequencing or next generation sequencing. So basically what it does is you chop up your DNA into little pieces, and then you can load it into this machine, and it it will replicate the DNA and like fluoresce in the color of the base, and then you can read it off. So anyway, this is an Illumina sequencer. It's the most common type of sequencing in the world. They have like 80% of the market. And then basically you get a file where it tells you what the bases of your DNA are. And then you can do the bioinformatics. So this part is the lab and the sequencing facility, and then come in at the end to do that. Okay. So before I get into sort of the more career side, any questions about all of that? Yeah. Yeah. All right. Okay. So I think this is, you know, a career talk. So I was explaining how you can become a bioinformatician, starting with how I did it. So, you know, when I was in high school, I decided that I wanted to major in science. Like, you know, that was my favorite subject. You know, I really couldn't see myself like going into another area. But I had no idea what bioinformatics was. I didn't know it existed. So this was definitely not the plan. So I did undergrad. I majored in biology. I worked in different professors' labs, you know, got a little bit of experience that way. Then after college, I couldn't get a job. So in science. So I was teaching math. And you know, I really wasn't happy with it. I like teaching, but not like 24/7. So I started doing like a lot of work. I started teaching myself stuff online, taking online courses, trying to figure out what I wanted. And then I really realized that I wanted to do bioinformatics. I've had one class as an undergrad. You can't, side note, you mostly can't major in bioinformatics as an undergraduate. I only know of one university that has it as a major. So I'll get to that in a second, but like everyone majored in like something different in undergrad. So as long as you're like in biology or related field or computer science or math, like you can wind up here. Anyway, so so going back, so I figured out that I wanted to do this, I thought, because, you know, if you think, it's hard to say, because if you think about like your biology class in high school, like there's no math in it really. And it's the same thing in undergrad. Like when you're getting a bachelor's degree, there's no math. It's not very computational. You do like a lot of lab work and a lot of learning, memorizing stuff. And that had always frustrated me. And that was sort of why I was thinking, oh, maybe I don't want to do that. But then I found this field and that worked out. So I went back to school. I had my master's degree in bioinformatics from Temple University, which again, this is like a really new field. I was only in the second cohort of this master's degree. The program had literally started the year before and no one had graduated yet. So
Kelsey, could I interject just for one more question?
Yeah.
You mentioned math.
Yeah.
I myself don't have a lot of, I look at my daughter's 8th grade math now and I'm like, I don't remember that. But how much, you must have had an interest in math going through high school and into your undergraduate, right? So maybe you could touch on the interest that you had in the math briefly, but then also like what math courses did you end up taking through college, not realizing that this was going to be your path?
Okay, well that's gonna be it's gonna have a kind of a weird answer. So I was always like good at math. Like I took calculus 2 in high school. Like
Oh wow.
So but I didn't think that I don't know. So but I didn't like think about like, I was like, I'm not gonna like major in math. Like what am I gonna do? Just sit around like solving problems all day? So I like haven't really thought about that. So for like, and then actually I took no math classes as an undergraduate because I had AP credit. And then in my master's degree, I took biostatistics. That was the only class. So I'm like my personal experience is kind of unusual. I would say the math that's most helpful for bioinformatics would be, well, probably you want like basic calculus, linear algebra, and biostatistics. I really wish I'd taken linear algebra and I never have. But like the thing is, like, day to day, you don't do that much, you don't like do math. Like, what you, as I'm getting into it more in like a minute, is like what you do is you do scripting. So, or you do programming. So like you write code to get the computer to do math for you. And I would say, like, if you've never written code, it's more like doing math than like writing a paragraph. It's like you're writing a series of like very specific commands to the computer. But like you mostly do that, not do math. Where you need to understand math is in order to understand what you're doing. You need to like, you need to know the background behind it. To try and give an example, I was asked to develop a new way to compare compare how methylation changes with age, which I have again in like a few slides later in the presentation. When that, it doesn't matter. I asked to find a new way to compare things. So I went and looked at different, I like went and looked at information theory and different like math techniques to find something that I thought would work for the scenario. And I also like a lot of reading like scientific papers and looking what other people did. So it's like you need that background to be able to understand what you're doing, but you don't like, there is no like actively sitting down and like, you know, writing down math equations.
And then your computer science experience, did that come after college as well?
Yeah, so I had, so about, you know, when I was trying to figure out what to do with my life, I started doing like a little bit of programming online courses, and then but I really learned in the master's degree, and I really like picked up what I needed in a couple of classes. And it's really amazing. Like I definitely, like, once you understand like the basics of how to program, it's easy to like learn more things. And I can't emphasize enough, like, once you start doing something every day, you learn to do it so quickly. Like when I started working as a bioinformatician, my scripting was terrible. Like it was really bad. Like I could get it to do what I wanted it to, the computer to do, but like not efficiently or elegantly. But if you do something every day, like you get better at it faster. All right. Yeah. All right. Yeah. So anyway, so went back, got a master's degree, totally changed what I was doing with my life. And then I got hired as a bioinformatician first at Temple, and then some of you might have heard when we were talking at the beginning of the call. And then our entire group moved to Coriell, and I've been at Coriell for like 2 years exactly. Yeah. Okay. But like I was like, so that's how I got there. But there's so many different ways you can get to the same job. And this is true in general and not just for bioinformatics, but I'm going to give you bioinformatics specific examples. You know, like I said, this is a really new field. Like really like the current way it's the, you know, the current practices for genomics have really only been around for 10 years. So like literally everybody I know got into the field in a different way with like a different path. So, and like these are all examples from like different people I know. You know, there's like people who started like majored in biology, got their PhDs, were lab scientists. They were doing bench work and experiments. And then they start doing more data analysis, more data analysis, more data analysis, switch over to working on a computer full time. You know, I know people who started as computer scientists and were working as programmers and decided that it wasn't like fulfilling enough and switched over to doing bioinformatics. So I know one guy who is has got an MD PhD, so you can do that at the same time. So basically, the way that works is or the way it worked in that program was you do your first 2 years of your medical degree, then you stop, you do a short PhD where you do a research project. And so this guy, he did bioinformatics research project, went back and then you go back, you finish up your like second 2 years of your MD with the clinical degree, and then now he has his own lab, does research, also treats patients. So this can be combined with a medical degree. It's just a lot of work. It's like 4 times the work. And I know people who started as neuroscientists, he was working in a lab, he was bored, he taught himself to code, became a bioinformatician. So there's many ways you can come to the same thing. Okay. So just trying to say, like, what does a typical day look like for me? What do I do? So I'd say the bulk is data analysis. Like, sitting in front of a computer, like I said, I'm like giving it commands to do stuff for me. There's a lot of teaching yourself new things, which is something I actually love about this job. Like, there's always, like, it's a fast moving field. Science is fast moving, and generally people are always discovering new things, developing new programs, developing new assays. For example, there was, like twice now, there have been like totally new ways of sequencing different things about the genome. And it was just like, well, here's the data, please analyze it for us. And I had to figure out how to process it. Which sounds intimidating, but I'm like, it's fun, it's a puzzle. So so to do that, we maintain our own servers. An individual like computer does not have enough processing power to do this sort of thing. So we have servers that we run stuff on. Do a lot of presenting, you do a lot of reading scientific literature, some meetings. Also, I get to teach other people bioinformatics. So I guess I could talk about that now a little bit. So at work, we have a group. At work, there's a lot of people who do like some bench work and some data analysis. So we have a group where they can get help, where we can teach them new things about bioinformatics and data analysis, so they can do it on their own. So that's part of when I wind up teaching. And then last year, because of COVID, Coriell normally has a summer experience, like a research summer experience for high school students and people who are early undergraduates, like the end of your freshman year or something like that. It's in person in the lab. Last summer, that wasn't happening. So we decided to do a bioinformatics research experience, like pivoted last minute. So that we decided to be for college students only, because you do need like a little bit more experience with like math and programming we were looking for. So basically what that does is it teaches you like the very basics of bioinformatics. It teaches you scripting in R, how to do RNA-seq, which is looking at what's being expressed in the cell. So it's like a 1 month, like really intensive course where we were on Zoom every day, teaching them something new in R every day, doing like a lot of scripting, a lot of programming, did a little bit of literature reading. Also, got lots of scientists at Coriell to, come give talks to the students who could see sort of what the possible career paths are. So I actually would encourage you guys, if you're interested, to apply for both the bioinformatics summer experience once you're in college, and we're not having the normal Coriell summer experience, but we're hoping to have it both of them in 2022. So if you're interested in bioinformatics or you're interested in working in the lab, I would encourage you to apply for one of them. And about like the different careers at Coriell, I'm like not the typical person at Coriell. Coriell is mainly a biobank, so we store samples that people have given for scientific research. It's mostly funded by the National Institutes of Health. And so I would say like 80% of people work on the biobank side at Coriell, although we're expanding research actively. Although, you know, COVID kind of ruined that a little bit too. It's made it difficult.
So, Kelsey, speaking of COVID, obviously your backdrop suggests that you're working from home.
Yeah.
In a career like this, working at a place like Coriell, was this something that you worked remotely prior to COVID, or were you more in the lab with the scientists working with them, and now you've just had to resort to Zoom meetings? And then do you see your work environment changing post COVID because of the work that you could do now sitting in front of your screen at home?
Okay. Yeah. So, oh, nice. So let me say about COVID. So yes, I have been working from home for almost a year. The year anniversary is in like 2 weeks. So anyone who did not have to be in the building was sent home a year ago, and I've been there once to get a flu shot, and that's it. As far as the lab people, Coriell quietly just started testing everyone. It's not official, but you know, we're a lab facility. The test is very straightforward. We had it up and running quickly. So that people who had to come in to work in the lab could be kept safe. And we have caught cases and had people quarantine. And so prior to COVID, I had the option to, you know, work from home 1 day a week if I wanted to. I didn't do it very much. I'll probably do it more after COVID because I'm more used to it. Also pre-COVID, my internet sucked and I had to upgrade. So that's made it easier. But yeah, I don't think it'll change very much for me. I know people are like saying that, like, you know, work from home is going to really change. You know, more people work from home post-COVID, but I don't really believe that. I think pretty much everyone will go back to the office.
Yeah, I mean, I bring that up because something that, you know, our first few speakers have talked about is that work-life balance. And, you know, especially there are a lot of young women on the call here from Gateway. So I think, you know, having that work-life balance can at times be slightly more challenging, I think, for a woman because of, you know, if they plan on having children and things like that. So, you know, I just asked that question because it's important for them to think about that moving through their professional journey and how you balance work and life together, and whether working from home. I didn't know the benefits of it until I started doing it. And now I never want to go back to an office or travel to, you know, jump in a car for 20 minutes, driving to Philadelphia again. So, you know, just wanted to,
yeah,
you know,
no, absolutely.
There was some background to my question there.
Yeah. And like, definitely this job has more flexibility than, like, you know, if you work in the lab, like, you can't work from home. You have to go into the lab. Like, you don't have a centrifuge in your laundry room. I will say, just on work-life balance. So the thing about bioinformatics is there's infinite questions you can answer. Like, literally, I could work, you know, 20 hours a day and like never run out of questions to answer. So like, you do have to set like boundaries for yourself. It's just like, you know, this is what I'm gonna do, like this and this day, and then when I'm done, I'm done. Like, it's like, it's like a weird, I think that might actually sound kind of weird compared to like working in a lab. When you compare yourself to other scientists in a lab have defined stopping points. It's like, well, this thing needs to run for 12 hours, so I'm going to go home. So bioinformatics doesn't have that defined stopping point. You have to like create it for yourself. Right. Oh yeah, where was I? So this is the last part for this section. So just about our group at Coriell, it's like a 15-person group. The number keeps fluctuating up and down as people leave for new things, and like we hire new people, which again has been challenging because of COVID. So our group studies cancer and aging, and the next section I'm going to try and give you some examples. And I really want to say that like bioinformatics doesn't exist without the people who work in the lab, the scientists and the technicians and the people who do all of the boring wet lab stuff that I don't like, to give me sequencing data. Yeah. So that is an old picture of our lab, yeah. Okay. So I'm just going to pause here for a second and see if anyone else has any questions. Drink water.
Anyone?
I guess, well, I had a question. How does this kind of relate, really? Is it similar to being a statistician for a hospital or healthcare organization, do you feel like? A little bit?
Yeah, a little bit. The data we work with like is really different, but
Right.
yeah, it's similar.
I have a quick question.
Sure.
What advice would you give to someone who is unsure, like what field that they should major in, like especially in school, like for colleges?
Okay, yeah, that's such a good question. So I think the number one piece of advice I would have is make sure you go to like a big school. Like go to a big university. Like if you're not sure, like you want to make sure you have options. You don't have to decide right away. Like if you go to a big school, you can, try some biology classes, try some history classes, try some, business classes, see what you like. While you're like you'll have gen ed requirements, so you can like fulfill them and like try stuff out at the same time. The thing is, if you go to like a really small, like sort of liberal arts school, you're more limited in the options you have. So I would say like you want like a university, one that has multiple colleges. And also you really want to pick a university that has good connections. And this is gonna sound like I didn't get this when I was in high school. So it's okay if you don't get it. But if you go to like a little college in like the middle of nowhere, if you don't pick the right major that has all of the good connections, like it'll be hard to find a job or like hard to move on to the next step. So for an example, because that's like really abstract, like I went to George Washington University for my undergraduate. So it's in Washington, D.C. It's really known for political science and for international affairs. I know it wasn't like the worst place I could have gone, but the biology department wasn't as strong, and like their connections to like local organizations and like other universities, because unfortunately, like in science, you really need advanced degrees. It wasn't that great. So I would say pick a big school, and like if you think like probably you what you don't want to do. Like I was like, I hate English, I'm never gonna be an English major. Like, so like pick a school that's like good at the things you think you might be interested in. Does that help?
Yeah, thank you.
Yeah, of course. Anyone else before I go on? Yeah. All right. Okay, so I'm going to actually show you some real data that our lab and that I have worked on. But first I have to give you some background because yeah, because it's just some stuff I have to explain so you understand what's going on. So everything I've been doing before has been talking about genetics. So like talking about like DNA is made up of the 4 bases. You can read the sequence. That's cool. Like I'm sure like everybody's taken biology, like you know about that. There's like a whole another layer called epigenetics, and that's what our lab focuses on. So, bad. So you know you have your DNA, and you know it's in a double helix, and you know that that's organized in the nucleus of the cell, right? So you know that DNA is coiled around histones, and it's, you know, organized into compact forms and like gets shoved into chromosomes when the cell divides, whatever, right? So it's organized in the nucleus. So epigenetics is the regulation of the DNA. So it's regulating the 3D spatial arrangement. It's regulating what gets transcribed into RNA and expressed. It's so epi is above. So it's above genetics. It's the regulatory layer. So what I'm going to be talking about is all DNA methylation. So there's actually on a cytosine, you can add something called a methyl group. And so you chemically modify the DNA base. So it's still a cytosine, but it has a tag on it saying I'm methylated. And so then what that tells the whole regulatory machinery of the cell is that mostly things with methyl groups on them are silenced. So it's, if you have the methyl group on it, it's being like, don't transcribe me into RNA. I'm just gonna be quiet and turned off. Because I actually forgot the one step, because when you think about it, so all the cells in your body have the same DNA, but they form, you know, hundreds, thousands of different cell types. So epigenetics was originally talked about in that context. So this is what like tells things to turn off in your skin cells so they don't become lung cells. So this is like the regulation that makes each cell function as it should, having its right cell type and its right identity and its right function. Okay. So I said a couple of slides ago that our group studies cancer and aging. So I have a cartoon. So the gray is your DNA, and then the blue is a gene that we want to, that this cell wants to have turned on. And then each gene has something called a promoter. So like the promoter is a place for like the transcription machinery to bind. And like if it's open, it'll transcribe it into RNA and then make it into a protein. Anyway, so the normal thing for like a gene is, so if you think of these lollipops as like methylation, so normally methylation is there in between genes, it's there in the gene body, and then it's not there in promoters that are supposed to be expressed. So if the promoter wasn't supposed to be expressed, it would be turned off. But it's like open, so it's like, I'm on, you can come and transcribe me. Okay, but as you age, this regulation gets messed up, like mistakes get made. So just like a couple at first, like really slowly. So, you know, maybe this one's on now, and then we've got a couple that are off, and then this one's on. And so like, this is fine, it can be tolerated, but in extreme cases, when you develop cancer, your methylation is really messed up. Like it basically, the pattern flip-flops. So like if this gene is an anti-cancer gene, well, its promoter is silenced, and so it's turned off. So basically the takeaway is methylation gets messed up as you age and as you develop cancer. Okay. So this is from a paper our group published several years ago. So this is about DNA methylation mistakes accumulating with age. So this is just a scatter plot. Everyone's seen them before. So if you look on the y-axis, this is the amount of methylation when in a young individual. So this is going to be measured from 0 to 100 for like reasons. So 0, like closer to 0 means you're like not methylated. 100 means you are. And then and then on the y-axis is the amount of methylation when you're old. And then this panel is in mice. So if you look at young mice and you look at like where the dots are, like the things that were not methylated, so that have low methylation when you're young, gain methylation as you get old. So you see like they're going up. And then things that are turned off when you're young that have methylation lose that as you get old. And not everything. Like there's still lots of things that are still off in both ages, but like quite a lot of places in the genome. So it's becoming dysregulated. Mistakes are getting made. So this is mouse. This is in monkeys. So you can see the same pattern. When you're young, when you're old, things that were unmethylated become methylated. Things that were methylated become unmethylated. So we're flip-flopping. And then the same thing in human, of course. And actually, so this study was looking at, we know this happens. Does this happen across species? And we looked at mouse, we looked at monkeys, we looked at humans, and the answer was yes. And the rate is actually consistent with your lifespan. So mice live 2, maybe 3 years. So they have a high rate of methylation errors per year. And then monkeys live longer. So the rate is lower, but like it's a consistent amount of change over the course of your lifespan. And then humans live the longest. So again, the rate is lower, but like you tend to accumulate the same number of mistakes over your lifespan. So that's one thing we look at, like looking at the errors that accumulate as you age. Another thing we study is cancer. So you may think of cancer. So I think some people think of cancer as a genetic disease where you accumulate mutations in your DNA yourself, and that 100% happens. But you also have epigenetic mistakes, and specifically methylation. So methylation will become altered in a lot of cancers. And so here you can, so these are like actual samples from people with cancer. This is leukemia, I think. And so you can see people are divided into 3 groups. So there's a red group with the lowest methylation. There's this blue group with sort of intermediate methylation, and then there's this orange group with very high methylation. And so the reason dividing them into these groups is important is there are chemotherapy drugs used to treat epigenetic effects in cancer. So so like this orange group, which is super highly methylated, would benefit from those drugs because they generally demethylate things, so that can help the cells reestablish normal function. Whereas, you know, if you're less methylated, then you won't benefit from those drugs. So like one of the things we're looking at is like which patients will benefit from this drug, which patients won't. Can we predict survival? Stuff like that. And you know, there's many different cancer types where you have different methylation phenotypes: colorectal cancer, leukemia, glioblastoma, stuff like that. And then just one more thing, looking
Kelsey,
at,
can I ask a quick question about the cancer and the species, just looking back at like the mouse versus the monkey and human data there?
Yeah.
my understanding of is that there are certain species of animals, I think it's like sharks and elephants, that really, it's been shown that they don't get cancer. And I was wondering if anybody has ever looked at a link between those animals' lack of cancer
Okay.
and other
Yeah.
types, epigenetic marks
No.
and things like that.
Yeah, so I mean, epigenetics is not as big a field as genetics, but it's getting there. So actually, yes, it's it's that paradox. I can't remember the name of the paradox right now. But actually, the thing is, if you're talking about age, mice don't get cancer. They just don't. Like, I know like lab mice get cancer, but that's because humans go to a lot of trouble to give lab mice cancer. Mice on their own like don't get cancer. Like very short-lived species don't get cancer. Intermediate species do. So like it's a problem for like humans, monkeys, dogs, whatever. Other dogs are domesticated, so they're weird. And then very long-lived species like whales and elephants and sharks, they like, people have found tumors in them, but it doesn't really seem to bother them. So as far as I know, no one's ever looked at like epigenetics and like that cancer effect. But like what we were saying in this paper is that like there's a steady rate of change as you age, and people have to look at DNA mutations in that cancer paradox. And there is a steady rate of DNA mutations as you age as well. And it doesn't seem to be related. The best theory I've ever heard for the cancer paradox is basically if you live a really short time, you die before you get cancer. And then after that, it's body mass. So like if a human has, a 1-pound tumor, that's really going to bother you. If a blue whale has a 1-pound tumor, like whatever, it's not a big deal for it. And the rate of like cell expansion and the cancers is kind of the same. So yeah, as far as I know, no one's really ever been able to answer that question. Okay. And last example, this is some of our most recent work. Just wanted to I think probably everybody's heard about the microbiome. Is it's not just your cells that belong to you in the body. You have like a gut microbiome, a skin microbiome that's made up of bacteria and small fungi existing in like mostly a peaceful coexistence with you. You're in a mutual relationship. But just saying, we're doing like some of the first studies looking at the relationship between the microbiome and epigenetics and methylation. We have collaborators in Florida who are experts on the microbiome side. So this first study was just really simple. It's like, well, does having a microbiome change your DNA methylation? So if you take mice that have been raised in a germ-free environment, they don't have a microbiome, they're just them. And if you take their intestines versus mice that have a normal microbiome, they're very different. Like there's crosstalk between the cells of your intestine and the microbiome. So if you look at this scatter plot here, so this is called a volcano plot, super common in genetics and epigenetics. So on the x-axis is the magnitude of the change in methylation. I seem to have lost my label. So like this site changes more than this site. And then on the y-axis is the statistical significance. So things that are like high and like in the corners are like the most interesting. But anyway, the point is we've got like about 12% of the sites we looked at that change, which is actually a lot. And then the interesting thing to sort of tie it in with the other two stories is when you compare age to cancer and when you compare the microbiome to cancer, it's the same locations that are involved. So, for example, we already knew this about age and cancer. I'm just using the picture from this study. Sites that change, like methylation that changes with age, is the methylation that becomes dysregulated in cancer. It's like 15 times, they're 15 times more likely to be the same places. And the effect is there for the microbiome in cancer. It's slightly lower, so it's like 4.1 times. I didn't have a good picture of it, but you know, some of these microbiome sites are also aging sites. Okay. So yeah, so that's some of what I work on. So,
Thank you.
Oh.
Students, do you have any specific questions about the bioinformatics field? Again, her career trajectory, and or some of that kind of interesting introduction to not just genetics that we learn about in the classroom, but the phenomena of epigenetics and how there's these additional regulatory processes in our cell that kind of affect the way our genes are expressed.
And I was just also add, if you have like questions about like, you know, college in general or grad school or anything like that, just please ask those too.
I thought it was really interesting. And I had also heard that one doctor said, sort of like the same point that David made, that basically everybody's going to get cancer if you live long enough. The opposite of like, if you live a short life, then you don't get cancer. But if you live long enough, eventually everyone gets it because your cells just kind of, you know, quit. and then they're susceptible to, you know, mutations.
That's really what the data supports. There are some exceptions. So childhood cancer, everyone thinks that you're born with it. There is, you know, an error in development, and you've had that cancer your entire life. There are some very, very strong DNA mutations that drive getting cancer early in life. The one everyone's heard of is BRCA1. If you're missing a copy of that, you're going to get, and you're female, you're going to get breast cancer in your like 20s or early 30s. But otherwise, yeah, cancer is a disease of aging. It's an accumulation of like small mistakes in DNA and the regulation of DNA over a lifetime. And yeah, pretty much if you don't die of something else, you'll like eventually get cancer.
And there's genetic and environmental effects to you know,
oh yeah, yeah, absolutely,
how quickly you might get it.
It's yeah. Oh, and just like one thing about genetics is like, drives me crazy how they teach genetics in high school with like Punnett squares and everything. That's like not how it works at all. Like pretty much like anything that's in your DNA is like also is influenced by multiple factors in your DNA, and is also influenced by the environment. Like almost everything is a combination of genetics and environment. And you can like help yourself out by changing your environment.
Do you look at like multiple, like all the different types of cancer, or do you only look at specific?
Rebecca, I couldn't hear the question. I'm sorry. So could you get a little bit closer to the computer, or just say it again, please?
Do you look at different types of cancer, like all the different types of cancer, or do you only look at like specific types of cancer?
Great question.
Yeah, so I don't make project decisions, so you know, it's whatever gets handed to me. But so not all the different types of cancer, because there's a lot. So our group focuses mainly on colon cancer and leukemia, so that's the majority. But I've also done some work on bladder cancer. Yeah.
What's the most interesting thing you've worked on, or aha moment that you had?
Oh, that's a hard question.
I'm all about the aha.
Yeah, I don't know if there's not like a,
or exciting, or you figured something out, you know, with some of the data that.
So okay, there's a lot of like aha moments or like really rewarding moments like day to day. Like I don't think I did a very good job of describing this, but like working this field is like solving lots of little puzzles every day. Like when you're coding, it's like so computers are really dumb, like really dumb. They're dumb and literal. So when you're writing code, if you miss a comma or you put a space in the wrong spot or you like misspell something, like it's not gonna work. Like I broke an entire website by having an extra S on one variable. Computers are really dumb. So there's a lot of like troubleshooting, and I actually like it, which sounds weird, but it's just like a puzzle to solve. It's like, why isn't it working? And there's a lot of like puzzles to solve with your data. It's like, okay, well, why is this weird? Why can I make it look like this? How do I reshape it? How do I bring in this other data? How do I compare it to this? How do I compare it to that? Like, there's just a lot of puzzles to solve. And so there's a lot of like, oh yes, I solved the puzzle moments. I think in science, there's not like a lot of like on like the larger scale of the project, there's not a lot of like aha moments. It's like the data is like so messy and it's not clear, and it's like really hard to be sure of things unless you're looking at them like, 10 years in hindsight when other people have like validated your work. Which I don't want that to sound like negative. It's just like, it's just like, okay, I think this is good. We're gonna publish now. All right.
I mean, I think in science, we hear about the major breakthroughs like E equals MC squared and the theory of relativity and the structure of DNA back in the 50s. There are a lot of science is just incremental. But those aha moments and those exciting moments, at least for me, came when, I got data that supported like my hypothesis for the first time, or really connected the pieces, and I got data that answered a question that I was looking to solve. I wasn't curing cancer at the time, but it still feels great when your experiments work and you can collect interpretable data that allows you to ask more questions and allows you to take the next step in completing that, what we often like to call these stories that we tell in science. When you write a manuscript or a paper, you have a collection of data which you hope is telling a story to your audience, to the rest of the scientific community, that helps to incrementally further that field. So, collecting those pieces of data and seeing how they all tie in together into this story, for me, that was one of the more gratifying pieces, I think, of science, and thinking about it as the larger picture, not the granular aspects of each individual experiment that you're doing.
Yeah, that's a great way of describing it. I totally agree. Yeah, I'm not curing cancer either. I'm hoping that I'm helping to push it forward. So like, in 10, 20 years, there's like a new chemotherapy drug approved. Like, it's not like, you're not going to get like the reward day to day of like, this patient is cured, but there's other rewards that are really gratifying. Yeah.
Well, students, can we give Kelsey a virtual round of applause here? Thank you for joining us today. Again, I think or I'm able to provide Kelsey's email address. You can reach out to her directly if you have any additional questions. And we hope to see you all again next week.
Thank you, Kelsey. It was a pleasure meeting you. It was really interesting. I learned a lot. You guys learn a lot? I understood. Awesome. I understood 75% of it.
Oh, good.
That's a win.
That's a win.
I'm at a C level, like an average C.
Oh no, that's good. I was so worried that it was not going to be that I was hitting the right level, because like you don't talk about epigenetics in high school.
No, and these kids are smarter than me, so they probably picked up on more, maybe 90%, right, girls? 90%. Becca's laughing.
Well, thanks again, everyone. Have a great day.
Take care.
Bye-bye.
All right.
Thanks, Kelsey. I'll follow up.
Thank you
All
very
right,
much.
it sounds great. Thank you.
Awesome.
All right,
Very good.
bye.
Sorry I turned off my camera, but I was having glitches
Oh.
going on. And plus then I was eating my lunch. I got
Which I need to go do
no,
now.
I
Yeah.
don't care. That's fine. Yeah.
Thank you. Take care.
All right,
Bye.
thanks.